Thank you. So, so I'm going to tell you quite general things about modeling. It's not so much about my research, even though I will mention some of the stuff that I have done in the past, but it's more about telling you about different kind of modeling you can do and what do they mean, and what they are used for. So I'm not going to show you anything that moves like Alex did. So it's quite boring, and I know you're hungry, but hopefully these things together can make it more salient for you. So just generally, I want to basically pose the question for you that what is a model and what do we mean by computational models? So since I just want people to be on the same page, I'm not talking about fashion models or animal models, but it's just more about computational models. So can you tell me, like, what do you think about models? Like when I talk about computational models, what does it remind you? Of course, you think about what Alex showed you, which is okay. It's also kind of modeling. I will get to that. But what is more general you think about models in psychology or neuroscience? What does it remind you? Anybody? Yes?
How the brain responds to the world in some quantifiable way.
Okay. So quantify how the making models to quantify how the brain responds to the world. Yes.
So like a mathematical description of a behavior or cognition.
Uh-huh. Okay. Yes.
A framework from which you can make predictions.
Okay. Yes.
Something you use to generate data that will tell you how more visible to be.
Perfect. Okay. Good. So you already know most of the important component for modeling. But just one thing that you should always remember is that even though now more and more we are moving to our computational modeling, people have used models because you cannot think about the brain or behavior without assigning certain regularities for it. So people always had models when thinking about the brain. And just mention a few things about computational modeling, and it's mostly about from the paper I sent. I don't know you got it or not. I'm not a good slacker, so I don't know. I posted in the wrong place, or it's possible. But if you haven't seen it, you could go download that paper. It's a very nice paper. It's actually 10 years old. I couldn't believe. I read it yesterday, so something must have happened. But it's 2008. But the point is that, as you mentioned, if you want to go from models that are just something in your head, the way you're thinking about behavior or neural representation of the brain, to something that is more look like a model, you need to formulate it. You need to write down equations. And what equation gives you is that you will be precise, complete, and self-consistent. So you cannot contradict yourself. While if you use word models that people still use in psychology, you can contradict yourself all the time, and there's no problem with it. So the other thing about advantage or use of modeling is that you can formulate, explore, and reject models at a much faster pace than experimentalists can test an idea or hypothesis. So that's another thing that if you become good at modeling, you can achieve, you can outpace experimentalists. And it's okay if you're an experimentalist too. You can outpace yourself. So, and then one person mentioned here is that for theory or modeling to generate ideas prior to experiments. So and so we usually do that. Again, you have a model of how things work and you design an experiment. But if you go back and look carefully about what you had, and when you get the data, not apart from the fact that when you get data, you say, oh shit, how it has anything to do with the hypothesis I had. But you realize that there's so many things that you couldn't even think about it in your model because your model was not formulated. But if you have a computational model before going to the data, you have many ways of looking at the data and making predictions and test them actually. And which brings me to this point that Larry Abbott mentioned very clearly, which is the difference between predictions and postdictions. So in all natural sciences, people talk about prediction. And it's really important. You generate the model, you make prediction, you go test it in the experiment. But really what happens, really, is that everybody does postdiction, which is you have some data and you explain with your model. All the models we know, they're inspired by certain data, experimental data. And then when you formulate a new model, basically you try to explain many of the previous data. And it's rarely that actually you come up with predictions. that are really, really new, and nobody has thought about them before, and then you go test it. But as Larry mentioned here, and I'm quoting him directly, is that there is nothing wrong with it. The point is that if you can use these postdictions to generalize to other situations. But as somebody mentioned here, it's really important to provide predictions, because that is how you can actually improve your models. So that's another important thing here, which is if you start with a model, go through an experiment, then you have a chance to improve it after you're done with your experiment. And that is how improvement can happen. If your model doesn't change from before and after an experiment, your model is probably wrong, or you're a genius. So you can pick it. But usually, because we have so much, we don't know so much about the brain, it's really improbable that you have a right model. So you basically, you want to always look carefully and see how you can improve your models based on the data. So don't feel shy about changing your model and saying that you were wrong at the beginning. So this thing, I think this is maybe more important if for different types of computational modeling that people can do, and people do, is that, so one type is that you use basic principle to understand what is going on in the brain. So basic principle that people have used, example is, for example, efficient coding hypothesis, that the brain tried to minimize the number of spikes to carry certain amount of information. You can think about Bayesian inference. It's a principle that you can use this kind of Bayesian inference. That's how the brain performs computation, and you try to map it into brain and understand what happens in terms of behavior or neural activity, or in general, you think about optimality. There are many notions of what is optimal in terms of neural processing or behavior that can guide you in understanding the behavior you observe. Because again, if you have, and this is, this group is concerned with naturalistic stimuli and experiments and so on. When you go into the experiment, usually it's very difficult to figure out what is going to happen. So you can use this normative or basic normative models, or these basic principles, to basically figure out what would be the ideal case. What is the best thing you can do, and then use that as a reference for what you observe. These are, I would call them prescriptive models, because they prescribe what should happen. Based on some principles, they prescribe what should happen. They tell you this is how the subject should behave. This is how the neural response should be. Okay? But again, you can use them to explain what happens. And then, so one limitation of this model is that basically the hypothesis is very clear. Yeah, so predictions are not really, these are not making predictions, because you can't even update these models. But then there are different types of models. Again, they have different uses, and they're all important for understanding how the brain works. And these are the neural models that people built at different levels, and I show you some example about what and how of neural responses. So you can have a model that describes the relationship between neural response and the stimuli that evoke them, and basically tells you what a system is doing. So all the stuff that Alex told you for the past 1 hour falls into this kind of models. Basically, you try to look at the neural activity and a stimuli, in that case was natural stimuli, to tell you what the system is doing, how is it processing information, what is related to what, which part of the brain is doing what, and so on. So these, you can call them descriptive models, because they describe your neural data or your behavior. And again, if you think about it, they have a different function than the prescriptive models, yes? You can update them, you can change them over time, and better understand what the system is doing. But they don't tell you how the system is doing it. For that, you need some different kind of model, which we call mechanistic models. And they can be circuit level models, or it can be at the lower level. It can be at the synaptic level. And they basically try to account for what you observe, the features you observe, physiological features you observe, and how the system works. So they allow you to understand or extrapolate how the system does it. So if you want to understand, why you observe certain patterns of similarity in the brain, then you need to go make a model at the circuit level to generate that. And then basically, if you can generate it, then you can say, I think this is probably what is happening. And I give you an example to be more concrete. Are these distinctions between different models clear for you? I just want to give you a guideline that when next time somebody tells you about computational modeling, which you hear all the time, try to think about what it means, what they try to achieve with that model. Because there's a danger here, which is people often use these descriptive models. And even though they are describing something, and Alex is smiling, and then try to basically tell you how the brain works. Okay? And a very good example, or a very bad example of it is my favorite bad example is drift diffusion model. Okay? So drift diffusion model was invented to describe, basically, what is happening in terms of reaction time and performance. Yes? And it describes, basically tell you this is probably, because that was the origin, yes? To give you a measure for reaction time and performance at the same time that you couldn't get with the previous models, that they didn't have that dynamics in them. But then later on, people shamelessly start actually saying that this is actually not a description. It's actually a mechanism for how the brain does it. When they map that map that model to the brain and say, look, this is the brain area, this is the neural activity, does exactly that thing. And then suddenly turn to a mechanistic model while you ask, wait a second, how does the brain can integrate information over infinite time scale? Okay? And then suddenly they say, no, this is just a description. So I hope, I'm pretty sure somebody here get offended by this statement, but I'm pretty sure you know somebody who will get offended by that. But anyway, so this is something you want to always remember, that when you describe something is different from saying how it happens. If you want to know how things happen in the brain, you need to look at mechanistic models. Is it clear? And I give you an example of a descriptive model and mechanistic models, so it becomes more clear what I'm saying. Any questions? Yes?
What's a basic principle?
So again, the basic principle can be something like a few examples there, but the basic principle is that brain wants to perform this computation this way. Why? Because this is optimal. Okay? This is the basic principle that should guide everything. Let's say, I would say everything you do is optimal. You have been evolved to be optimal in let's say foraging for food. And then I say, okay, if I leave you here in this environment, what would you do optimally? And then I can characterize this should be your behavior, and I tell you, okay, you have to go to that chair directly because that's optimal. And then I check with your behavior and see, okay, how far you are from that optimal behavior. So just assume that the system should work like that in a certain way. So a better example is efficient coding hypothesis, because it has driven a lot of results in theoretical and computational neuroscience, trying to explain why neurons respond the way they respond. Because there is nothing about that, like, you know, why let's say a neuron in V1 should respond this way. And they try to say, oh, the way they do it is because they can maximize the information they can transmit with the lowest number of spikes. Why is it important? You can say, oh, because the brain is about transferring information, and but at the same time, you don't want to consume so much energy. So that is the principle that probably have guided the evolution of the brain, and then you test it. And you could be wrong in your basic principle. Yes? Artificial neural networks are, in a way, they are mechanistic, because they're saying that how the computation happens. But then when you have and then people map it into, like, you know, different areas in the brain, so you can say that they're providing a mechanism. But when you look carefully at them and you say, okay, are these mechanisms realistic or plausible? Then you can, you know, ask but you can say they are mechanistic. All neural networks are in a way mechanistic. But then you can ask, the mechanism that they provide is plausible or not? And then, so whether those models are useful for understanding something about the brain or not is about the mechanism they provide is useful. Descriptive model, you can ask, you know, is the description that they provide is closely matches what happened or not? Yeah, we can qualify them or verify them like that. Yes, but at the same, they tell you, like, you know, if you do this type of convolution, they can tell you how you can actually connect them. That's basic. So all the mechanistic models also do connect things. Otherwise, they're another model. So they say, if I assume certain things here, write these equations down, this model is going to behave exactly like the V1 you run in your brain. So at that level, they have to match what you see, but then they tell you how it happens. That's something that the descriptive models don't tell you. Yes?
What do you think about like reinforcement learning models? Is it also a deviant situation?
Yeah, the reinforcement learning models are not mechanistic per se, because you just write down equations and say this is how you update things over time, like you observe a feedback and so on, but it doesn't tell you how. Yes. So and then you say, oh look, I described the behavior over past few hundred trials, and I can closely match what the subject is going to do or what they have learned. So you are really describing the behavior more than explaining how it's happening. But then you can go one level further and say, this update rule that you have found, how it can happen in the brain. And then at that level, you can call it mechanistic. There is a very fine line between the two, and that's why people go back and forth between the two. Just want to be clear when you say something, but we reveal the mechanism for something. Ask yourself, is your what you're suggesting can be a real mechanism, or or just an equations that you have written down. Okay. What time did I start? At 11:45 or no? Okay. Now I just wanted to ask because I haven't it's just the beginning. So what is a good model? This is again, these are mostly Larry Abbott's words, and I agree with them almost entirely. He's one of the best computational theoretical neuroscientists I know. So a good model, the important thing is that to find the minimum set of features that basically allows you to explain certain data. And there is something here dangerous here, what we call realistic models. And I have used that word before. Everybody does it, but the point is very clear by this saying from Borges about the basically map of the empire that was the same scale as the empire and coincided with it point for point. Yep. So you can make a very complex model that exactly has all the details that you can imagine as the data. It's kind of possible right now because of the data that people have right now. But if your model is complex as the complexity of the data, you gain nothing because you can't study it. And an example of it is like Blue Brain project. Hopefully there's nobody from the Blue Brain project here, but basically the goal is to simulate the entire column of cortex, yes, based on all the data they have. And then you end up with a model that is very, very complex because it has all the complexities. How useful is that model? Not so much, because you cannot figure out the features that are important for generating that data. So when you think about realistic models or building any model, ask yourself a question about what kind of data you have and what do you want to achieve and test, because that's basically should be your guideline. So basically, as Larry says, the art of modeling is what subset of features are important to explain your data, and don't go beyond that because you cannot verify it and you cannot use them properly. And the complexity of the model does not allow you actually to interpret the result of your models or simulations. Yes?
So on the one hand, it seems like you're arguing that maybe the question that you're asking should determine what level to model and the complexity of the model.
Yes.
On the other hand, you could say that the complexity of the model necessarily kind of limits how generalizable the model can
Yes.
be to do
Yeah.
things. So,
Yes.
I don't know if you have any insights.
So that's exactly, so the way you do modeling is very similar to do experiments, yes? You have a big idea, you test it, and then you go into details of it. So you make a model that could be more general and doesn't have so much details. You test it, and then you refine your model constantly. Yes, that's a process that you have to go through constantly in order to figure out what you need to add to your model to generate new details that you can test. Of course, you can always add a lot of details that you cannot test with your models, and that's not useful. But yes, it's a process, it's a recursive process that you have to go through in order to figure out what are the features that you need to have in your models. And again, I will give some examples so it becomes more clear, hopefully. what I mean. But that's something that you have to learn over time about the level of details that you need to have in your models. And that's another part of the modeling, is that testing different implications of the features you have. So you have some assumption, you have some details in your models, and the good thing about modeling is that you can turn them on and off and see what happens. So again, let's say if you are trying to simulate some cognitive behavior, and then you have in your model something very specific about short-term depression at the synaptic level in some neurons in your model, and you turn it on and off, and it has nothing to do with the behavior you see, behavioral or neural activity that you can observe. So what is the use for that? Not clear at the moment. You can say it would be important for later on, or if you want to make prediction about how drugs, let's say, influence that types of synaptic depression mechanism and how it relates to cognition, you can do that. You can make predictions with your model and then test it. Again, think about what you can do experimentally so that you can advance your models. Okay, so what I'm going to do for the rest of the talk, if there's no question, is to give you a few examples of an example of a descriptive model. It's called the saliency model. Some of you probably know about. And then some experimental evidence about the formation of saliency signals in the brain, and show you two examples of mechanistic models from my own work about how you can ask questions about saliency computation in the brain. Again, here the concern is that we want to know, this is the only naturalistic scene I have here, okay? So why visual attention or bottom-up attention is important is basically when you're looking at the scene like this, what guides your eye movement. So we move our eyes all the time in order to process information differently, because wherever you foveate, you have better resolution. And the question is that what in this image guides your eye movement? So that's the question about how bottom-up attention influence your saccades. The question that people ask is that what is really salient here? Is this wildebeest kid, wildebeest here is salient, or something else is a mountain, etc. And they came up with different models over many years to figure out what is the most salient location. And the best model, at least 20 years ago, still I think is one of the best models, was the saliency model by E.T. and Koch. So in this model, basically, so you have this image, and again, you want to find the most salient location, which you basically want to use to describe, if you show this image to different people, where they are going to actually look at. Yes, that's the question. So you want to describe their eye movement behavior. So I tell you quickly, basically, what this model does is basically use the image and it decompose it into different features like color, intensity, orientation, and so on, and then perform certain computation on them, and it comes up with a saliency map which tells you what are the salient locations in the image, which means where people are going to look at if you show them this picture. So what computations are required here? Again, so start with this image, you look at different features, you do basically progressive low-pass filtering and subsampling of the image at 8 different scales. Makes complete sense, yes? So what you do is that basically you have this image, you have a lower resolution of that by filtering it, basically a lower resolution, and so on. Because what you want to achieve is that there are some things that are salient here that are really big, and there could be something salient that is very small. So you don't want to lose any of them. So that's why you need to do that. So they do it over different scales. The scale again means the resolution of the image. Then they do filter with different filters. So here there are Gabor patches that you can use to detect orientation in the picture. So you do that, basically you get this kind of maps at different scales again, and then you combine them. You perform what is called center surround computation. So you have a filtered image at a higher scale, and you subtract from from it a filtered image at the lower scale so that you can see the contrast between the two. Again, I don't want to go into detail of it. I just want to give you the flavor of the computation that is done in the saliency model. And there's a toolbox for it, and some of you probably have used it to figure out, let's say, when you show natural stimuli to the subject, where they are, what are the important things in the scene that you need to know about, because this model does a good job in predicting those points. So and then basically based on this computation, for each feature, you generate this conspicuity map, which tells you what are important in that feature. So in terms of luminance, these are the important things. Orientation, these are the important things. And the color, this is the map. And then they perform this lateral inhibition and normalization, basically to get rid of the noise. So you suppress the signal and the surround, and then you perform this successively to come up, and then you combine this features map into the conspicuity, into a single conspicuity map, and then you have the saliency map. So again, if you want to read about details of this model, you can look at those papers that are cited there, or just look at the E.T. and Koch saliency model. Okay, so just want to summarize what you need to do. So again, this model trying to describe where people are going to look at, what are the hot points, what are the salient point in an image. And it basically tells you this is what you need to do. You need to perform all these computations. Okay, so you may ask, what is this model good for? Yep. Again, what you want to get out of it is not trying to understand the brain. Yes, it's trying to understand the behavior in terms of saliency of the points or objects or spatial locations. Okay, so you can learn still a few things from this model. Is that maybe the main computation that you need to do, your brain has to do to figure out, oh, this is the most salient things that I need to look at and process, is this basically iterative center surround filtering and normalization. But then you may ask, how does the brain does this kind of complex computation? And also tells you there's a saliency map that you have to combine everything, and probably there are some places that there are this conspicuity map that are related to certain features. So because these are the assumptions of the model. You can say, my model is such a good description of the behavior, there must be somewhere in the brain that has some of the component of the model. Yeah? And then I can basically say, go into the realm of this is the mechanism that is happening. Yes? There must be a center surround some sort of computation that you can also do it with lateral excitation inhibition that gives you that computation you need. And then you can say, oh, I'm going to look for that, look at the brain to see where this computation happens. Because this is the computation my model actually does to get the behavior, yes? So it's a simple logic, yes? And then another one is that, this model completely separates different features, process them, and then combine them, okay? So you can ask, is it actually possible, realistic, or the features are completely separate in the brain, like color is processed completely separately from orientation, from intensity, to arrive to the saliency map. These are the questions you can ask. So to give you an example of, okay, so you have this model. Now you say, I really want to know, okay, this is great. It's a good description of the behavior, but I really want to know what happens in the brain. Yep, for some weird reason, you actually want to know the mechanism that is actually using the brain to perform these computations. So before that, I forgot to give you an example of the saliency signal in the brain. So one way of to look at the saliency signal is that if I show you this image, this is this thing called a pop-out. Yes, this bar has different orientation and color than the rest of them. And if you look at this image, it suddenly pops out. Yep, and that is basically, I can tell this has the saliency signal. This is salient location. And so people trying to look for the saliency signal in the brain, they use this kind of stimuli in monkeys and they look at V1. I give an example from different brain areas, visual areas. So basically, if you show V1, this kind of stimuli, so this is a single target, this is a homogeneous target, this is a pop-out target, and this is a target that has nothing, then they show, look, you can actually see a difference between these two responses, which can tell you maybe V1 can tell you the salient location. Maybe has this contributes to saliency computation, or actually has the saliency map. And there are some people actually still claiming that, but that's wrong. But because this is really old. So the better question is that actually, if you want to know the issue with that, just thinking about whether you have a pop-out versus nothing, is that it could be just, this is not telling you this is salient, this is telling you there's something here or not. You really want to compare these two kinds of target. So there is the same target here, the same green oriented bar here, but this is what's called conjunction target, which means that in order to find this in this, you need to know that it's different in both color and orientation from the rest. So this is a special target here, a special bar here, but it's not salient. It's the same guy here, but the surrounds are different. So if you want to know really, the argument is that if you really want to know where is the signal about saliency, you need to compare the response to these two stimuli. Make sense? People have done that. This is not the experiment like 20 years ago, 15 years ago in V1. And then I'm not going to tell you the craziness here, but you can conclude that it's not happening in V1 because the response to this conjunction and pop-out are the same in V1. Then people look at the higher visual areas before, and then there you can actually see a difference between the response to conjunction and pop-out, which tells you V4 has some saliency signal in it. And you can see how it emerges over time. It doesn't show up right away. It takes in a few hundred milliseconds that you can see different response to the pop-out and conjunction target. And people have used similar ideas using actually the saliency model, looking at fMRI data, trying to find out where it could be. And these are the results. You also can look at the change in the saliency signal over visual areas. And you can see that the signal gets stronger and stronger as you go from early visual areas to higher visual areas. So again, when you think about the saliency model, you can say, okay, you know, that's possible because you have this iterative process that goes, and this could be mapped into different visual areas. So you have some signal which is weak in V1 and it gets stronger as you go to higher visual areas. So here comes the mechanistic models. So if you ask the question of how this happens in the brain, yes? So you have this signal, seems that it's different in different visual areas, and you ask the question again, if you're really bored with your life and say, okay, where it's happening, then you can ask the question and then go build the model for it to try to explain the same process and say, okay, my model can replicate this data. It's possible that it happens through this mechanism. So I show you a firing rate model, and then I give you a more complicated example. So firing rate model, how you do that? So if you want to make a network of firing rate model, means that basically you're approximating neuron or a population of neurons with an equation like this, which is R is the rate, and this is a time constant for it, and there's a function that tells you how the input is going to drive this cell. Okay? Again, it's called firing rate because it only simulates the firing rate of neurons. It doesn't care about the spiking and so on. And you see there's not so much detail here. This is a parameter of the model, and this function also is a kind of parameter of your model. And then you can connect these guys together. Say, okay, if this neuron fires, how much current you are going to get another neuron. And then basically you have to simulate a bunch of equations like this. But what I want you to notice is that what you have is that you have this function, the weight of connections, and time constants. These are the parameters of your model. Okay? Again, the details are not really important. You can go read about firing rate models. It's just what you put in them are these things. They are not going to come out of your assumption. They're not going to give you something extra. It's just that these are the details you have in your model. And then you assume, you know, some neurons are connected to others through excitatory connection and inhibitory connections, but there's no detail about it. It all went to this weight function, if it's positive or negative. So what you can achieve with that, you can basically make this kind of model, which I tell you, you have a network of neurons connected to each other through, you have excitatory and inhibitory neurons with these equations, and then you can build a layer, you call it V1 and V2 and V4, and you connect it certain ways, okay? Again, details are not important. What you can achieve is that, so you can say, okay, if I show this stimuli, these different stimuli to this network, what would be the response of neurons in different visual areas that I just made up with this fine rate model. Okay? Again, it's a model trying to mimic or simulate the fine rate of neurons based on certain assumption. And what I can say that, oh, look at this. What happens in V1, these are what you want to pay attention to, the little garbage here, is this difference between these three guys and this one. Okay? So here, it's not, you cannot distinguish the response to this from all these guys. Down here, you can see that this is smaller than them. That is the different response to pop out on conjunction. That is the saliency signal. So I can say, look, what I put here was that I put identical layers. They're connected to each other through inhibitory and excitatory neurons. The inhibitory connections are wider, but they're all Gaussian. And if I do that multiple times, this is what I get. I can replicate approximately. I can replicate what people have seen in the brain. So maybe that's all you need is this lateral excitation and inhibition in successive layers. But there are a few other things. So this model also assumes that different features are processed separately. And because it's a model, what I can do, I can mix them as much as I like. I can assume that here that the signals are mixed or mixed in higher visual, like in V2 or V4 of the model, and see what happens to the behavior of the model. And then if I do that, I can see that, oops, you can see that I'm going really fast. I can see that if I do it right at the beginning, I don't see the distinction between this response and these three. So I don't have the saliency signal. But if I do it at the end, it's OK. I get what I need. So basically, it tells you that neurons probably supporting saliency computation have individual feature selectivity. They don't have mixed selectivity. Probably for most of you here, you say, who cares, you know? But somebody, let's say, recording from monkeys, they may be interested in that question of, you know, are these neurons contributing or have mixed selectivity or not? And then you can answer, oh, my model predicts such a thing, and then they can test it. So there's another one. I already mentioned this. So you basically, this shows that with simulation that you want to mix signals later on if you want to mix them. So you want to keep them separate. If you mix signals for saliency computation, if you have neurons that are selective to both orientation and color and other things, you cannot compute the saliency signal. Okay? You will fail. So maybe you are completely wrong. Maybe that's not possible, but that's another prediction, is that of course, not all the visual neurons have, you know, individual feature selectivity, but those who are contributing to saliency computations have. That is the prediction. Yep. Nobody has tested that, by the way. So, and this is not really important. So, and then you can also test one more thing easily with this model, which is if you have this layer of neurons, and on top of that you have the saliency map, and send feedback to them, you can ask the question, where should you send your feedback? Should you send it to V1, to V2, to V4? Okay? And where is more useful? So this basically, this tells you that the later area you send it is more useful it is. And you say, oh, this is a prediction of the model, that you have to have attentional modulation in higher visual areas. And somebody tell you, oh, we already know that. Yeah? So that is really a post-diction. Yes, we know that the attentional signal in V1 are quite weak. They're strong in V2. They're strong in V4. Yeah? We don't know so much about V2, but definitely they're stronger in V4 than V1. So then you can say, see, this probably explains that if you have feedback, you want to send it to higher cortical area, because that's where you can get a better improvement in your signal. Okay? So that's another thing you can do. Now you can say, so basically, these are the kind of, again, you can ask, these are prediction or postdictions. You know, how much of this we already know, that, okay, that's basically all you need to do to perform saliency computation. Excitation inhibition at different scales, not mixing neurons with different selectivity, and feedback from higher visual areas that are encoding saliency map. You can say, all these predictions are boring. I want to know something more exciting about saliency computation in the brains. And I ask you, what else do you want to know? Actually, to see you are awake or not. What else do you want to know about saliency signal in the brain, or how it's computed? Anybody? so boring question. I know. I worked on it 10 years ago, but so anything like comes to your mind, like really out of boredom, like you want to know about the saliency computation, like something.
How it's affected by context.
Context. Okay, that's a good question. Like basically how the context is going to change all this computation. So what would you do if you want to study that? Since you said it. I didn't tell you that that's what's going to happen next.
That would change the goal or the target of an experiment and see how that modulates whatever saliency signal that you.
Okay, and then so let's say if you want to put like a goal directed, let's say you want to say okay, how the context or the goal of the subject is going to change all this computation, you could say, okay, I'm going to put another population of neurons here that are going to represent my goal. And I'm going to project it to this other neurons down here and see what happens to the computation. Yes, you can study that. And then if you formulate a good question, you can come up with a good model and then go really test it. But if you come up with a lousy question, basically all you have is that it's obvious. Yes, you can come up with something obvious that you would know without even modeling it. What else you want to know? Let's.
It seems like some pretty high-level things.
Okay.
Face versus animate figures in general, which presumably depend on all these features you want to record.
Exactly.
Are really important for salience.
Exactly.
It's a very fast time scales.
Yeah.
How does that?
Very good. How these are, and clearly I don't know the answer, but that's why it's a very good question. But people have added face detection to the saliency model because it's such a strong thing and it's completely different beast. So they had to actually amend it to the saliency model doing this.
That's a hack, right? It's
Exactly.
V2 doesn't compute faces or doesn't
Exactly. So they have a separate system that at the end is going to combine, face detection and how it's computed to the rest of it. Yes. But then you ask like how face processing is different and that, and I can say, wait a second. So all I have in my model is that, how the process happened, let's say for orientation map. And then I can think about is face processing is different from center surround computation orientation map. Is it can you do the same computation with the same kind of architecture? Isn't it too slow? And then if I want to ask the question, thanks for asking that, then I have to ask how the speed of computation here in my model is determined. Yeah, like how fast the signal emerge. And then if I go back to the model I started, I have a tau in it. Yeah, it's a tau for the fine rate model. I put it by hand. Yeah, it doesn't emerge from the model. It's a parameter of the model. I can say, oh, the face, the tau is different for the face. And then you say, what the hell, where do you get that? So how you can get the time constant of computation here is a question that you can ask. But you cannot answer the fine rate model because fine rate model already has the tau in it. So you want to see how else, what else you need to do. Then you spend a few years of your life, like I did, try to make a spiking network model because in a spiking network model, the speed of computations are not at, you cannot adjust them as you want. You have to rely on synaptic current, and synaptic currents have certain properties, and you can't just put any number you like there. Okay? And then you can answer a question of not only the speed of computation or how fast the signal can emerge, but also you can ask the question of how different types of receptors contribute to this. How different kinds of drugs or neuromodulator could contribute to the saliency computation. See, this is a different kind of question you can ask, and then you can go and torture yourself with building a model that has those kind of details, and then you come up with something new. That's why I'm saying it's a process. Yes.
So question. You're saying that as you get these models that are more complex, incorporate more information that lets you test all these interesting things. But you started off by saying that the hyper-realistic model is useless.
Yes, exactly.
I kind of disagree with that. I think that you're kind of moving in that direction here.
Yes, I completely agree. You move in that.
Stuff, you can break things, specific things,
Yeah.
into hyper-realistic models that you can't in a realistic world.
I completely agree. What I'm saying is that you want to progress toward that model by adding more details when you can test them and break them and see what happens. If you jump from here all the way to something up there that has all these components, has all different receptors, different types of neurons, different types of connections, then you cannot even make sense of the model. You can definitely break things. That is, again, the idea behind Blue Brain project, is that you can actually do experiment with that simulation. Yep, but you have to verify that, and it takes forever to do so. But then at the end, you know, it's a chicken and egg problem. But yes, you can say, if I make a very, very, very amazing detailed model of the brain, then I can do whatever I can do with a slice on it. Yes, I can measure things and see what happens. But the complexity of the model, I think, does not allow you to understand what is going on in terms of mechanism. Like I ask you, okay, tell me what kind of ion receptors are there, because you have 25 different kinds. Which one is more important? It takes you years to figure it out, because, and probably all of them are important. So there's a seminal work by Eve Marder, did at Brandeis, basically look at a very, very dumb system, very smart system of gustatory movement generator in crabs. Yes. So basically, the only thing this system needs to do is to generate a movement, a chewing movement, because crab doesn't have teeth, so they have to chew the food in their stomach. So what they found was that they have basically the few hundred neurons that generate this pattern, few hundred neurons, but they have 8 different kinds of ion channels, and they have connections. They know everything about them. So what they found is that for this simple system, that you can measure exactly, you know, what is the frequency of the rhythm they're observing, and what kind of ions or ion channels are important. You can actually make a model and then test to see what you need to get there. And then what they found is that there are multiple solutions to the same answer, because of course when you are in 8-dimensional space of having different ion channels, you could be here and generate that pattern, or you could be here and generate that pattern, and so on. So if you think about it like that, now think about a system of thousands and thousands of neurons. How many different ways you can generate it. So that's why I think you need to move slowly toward that more complex model. I'm not saying that we shouldn't make more complex and detailed models. I'm saying that we need to make them at the stages. So saying that, so how you can make a spiking network model is, again, it's a model. You decide how much detail you want to put in it. Don't listen to anybody, including your advisors. So, because, you know, I can tell my student, oh, we need all these details. Go have fun, yes? But I'm not simulating it, so it's very easy to do so. So you can basically say, oh, this is a typical dendrite in the cortex, so we need to simulate this. And then good luck, because simulating this neuron takes a year even. So, but you can say, oh, you know, all these garbage are not really important for my purposes. All I need to have is this neuron that have a soma, has dendrite and axons, and I can use an integrand fire neuron, or you can make it more complex. Again, you can choose different kind of model depending on what you want to answer. And then your equations will be different. And the equation for, basically, you will have a different sets of equation with different sets of parameters. That's all that happens, yes? So you go from one model to another one, that's what you get. And then, and just basically showing that you can simulate something with this lousy model that looks like real neuron. Okay, let me go faster. So, and you can have different types of connections. So that's the question here, was that how different receptors contribute to saliency computation. So I put different types of receptors here with different time constant that we know about those receptors. And then you can build a model, tune it, and you get this garbage. So, and it basically has, you know, thousands of neurons that you have to simulate it, and it's really painful. But nowadays, if you want to do these kind of things, there's simulators that you can actually use to make something like that. Do you have questions? Yes.
Sometimes when I think about models, and I agree with you that they're like faster to break and faster to test, I do though come back to the question, like, at what point are we studying the model, and at what point are we using the model to study cognition or a cognitive system?
It's completely fine if you study the model. That's what we do all the time. The reason is that if you don't, what is the alternative you have? You're looking at one way. So either your experiment is really boring, okay, and there is nothing exciting. There is one thing that is bigger than the other one. Well, what do you want to know about it? It's bigger. So we found X is bigger than Y. It means blah. or you have very complex pattern of response, yes, behavior or neural response. How do you make sense of it? That is the question. So the only way I think about to make sense of it is that you have a model as a guideline that tell you this is probably what should happen if these are the assumptions are true. And then you can use that to say, okay, what it happens, it has anything to do. That's why, you know, basic principles are important. People use them because they say it's so complex, everything is garbage. You really need to use some basic principle to make sense of it. And then some people would say, oh, you need to make a model for that. So studying model is completely fine because it actually can guide you how to look at your data. Okay, I need to go faster. Yes. No? Okay. So basically, you can, all those figures I show you, you can generate them with this more painful model, a spiking network model, and it tells you, you know, how the signal is generated, different brain areas. But because of the details I put in it, I can tell you something about the influence of NMDA receptors. That, of course, you don't give a damn about, but somebody does, okay? And then I can tell you, okay, what happens if you change the NMDA to AMPA receptor ratio in the model? And then I can predict that from all the simulation that NMDA is really important for saliency computation. If you have too much AMPA, you cannot perform saliency computation. Okay? So I would predict that areas that are important for saliency computation should have more NMDA than AMPA, or that the balance need to be toward NMDA. The intuition for it is very simple. You could say you didn't have to do all this torture. Yep, NMDA has longer time constant, and it can allow you to integrate information over longer time scales. But anyway, so, and if you basically change the AMPA to NMDA current ratio, you can mess up all the computation that you had here in your model, and that's your prediction. So it tells you certain things. Again, now I did everything with the fine rate model. I did with the spiking network model. Sorry, ouch. I just gave you new predictions here in addition to the fine rate model. Okay? Actually, I never made that fine rate model before the spiking network model. This is just for you. So everything I said before, I could say with the spiking network model. The reason was that who cares about fine rate model? Because the fine rate model is very similar to what the saliency computation model does. So it doesn't give me so much. I really want to know how neurons can perform this computation, considering how noisy spikes are. That was the reason to torture myself with this. So, and then actually, so this is actually a prediction. I didn't make it up, okay? It's in the paper, it's 2010. And then, so the two studies later on showed up that basically talk about the role of AMPA and NMDA in saliency computation. So you can say, okay, I didn't waste all my time. But did my prediction actually make these people to run this experiment? I doubt. Okay? They could have different incentive to do it. Yes, they could have some hypothesis to do it. But then if you use this model, you could ask more specific questions. Yeah? Because it makes more specific prediction. So what I want to tell you, I'm done, is that don't be afraid of making more specific prediction, making more detailed models, because you can actually falsify them and learn something. If you don't have that, you can never learn anything, period. Thank you. Yes?
So I assume from the first half of the talk, the point you're trying to make is that there's a reverse inference problem almost with modeling. Just because your model happens to make the figure exactly doesn't mean it's doing the exact same the way the brain's doing it.
Yes.
So like, I'm going to scan the cat doesn't mean that's how the brain's scanning the cat.
Exactly. So that's exactly now. The power of modeling is that you can make prediction, say, you know, this is how it does it. And I say, these components are important. Now, if I break this thing in my model, it will break the computation. And then if I'm right, you go break that in the experiment, then you can see that, yes, you're right. That was really important for this computation. So probably you're right. Again, I could be wrong because the model has some level of abstraction, yes? You may say again, whatever I put in the model, I get out of it. The level of details I put in the model could be okay for the prediction I want to get and for the test that I'm going to have. So you can say, oh, you're just talking about NMDA and AMPA receptors. How about the interaction of them with something, yes? And I don't have it in my model, so I cannot make a prediction about it. And if you are really concerned about that, then you say, okay, let's make a model that has those details and then test it. But yes, That's exactly. You have the reverse inference problem, but you have a way to test it. That's what you get with having mechanistic models. Okay. Thank you.